Why astrobiologists are looking for purple planets. The universe is a harsh, brutal place. Solar winds, high radiation, and extreme temperatures and pressures make it largely inhospitable. And yet, life found a way. It's in you, and in me. And perhaps there is no quest more epic than the search for life beyond our beautiful blue home. But scanning the night sky for possible neighbors, like most things in astronomy, comes with its challenges. Where could they be? What would they look like? Should we even look for them? Are they, in turn, looking for us? One thing that makes the search for alien life tricky is that we don't know exactly what we're looking for. But by studying ancient life forms on Earth, scientists can get a clearer idea of the kinds of organisms that are most likely to be out there and where we might be best off looking. Join me today as we uncover how scientists actually hunt for alien life, the unique characteristics of Earth's earliest life forms, and the surprising role these ancient microbes play in guiding the quest to answer the eternal question: is there anybody out there? If you were tasked with combing through a seemingly infinite universe for signs of life, where would you start? I reckon I'd start by looking for a planet similar to ours. Starting the search in the most hospitable part of the galaxy seems like a smart move. The galactic habitable zone is the region of a galaxy that has the most optimal conditions for potentially life-bearing planets to develop. Typically, it's a spot with enough heavy elements for Earth-like planets and minimal cosmic drama, like supernova or stellar close calls. This excludes stars too close to the center or in the spiral arms of galaxies, since they're full of super intense, harmful radiation. So far, we've discovered 5,539 exoplanets of all different shapes, sizes, and compositions. Scientists are still debating what kind of atmosphere might be hiding signs of life on other planets. As far as we know, life on Earth emerged 3.8 billion years ago. At that time, our atmosphere was mainly nitrogen gas, carbon dioxide, and water vapor. You then understand why some scientists choose this atmospheric composition as a preferred focal point, but others aren't so convinced. They say only searching for these compounds is limiting, since methane or hydrogen gas atmospheres may themselves be signs of life. In reality, it seems to be a bit of a chicken-egg situation. The atmospheric composition of a planet determines what kind of life, if any, could emerge on that planet. But the kind of life that emerges also affects the composition of the atmosphere. Let me quickly explain with an example from home. Today, Earth's atmosphere is 21% oxygen. That wasn't always the case. It wasn't until about 2.4 billion years ago that free oxygen gas started accumulating in our atmosphere. This is known as the Great Oxidation Event. The main theory among researchers is that cyanobacteria living in the ocean evolved the ability to photosynthesize and started releasing oxygen gas faster than they could react with other compounds. Eventually, this oxygen evaporated from the oceans into the atmosphere, replacing the methane that was already there. This is an example of a biosignature of life, the next step in honing our mission to find alien life. Biosignatures are substances, signals, or patterns that could be a sign of biological activity. This could be something as obvious and direct as a fossil, or something more subtle, like the composition of a planet's atmosphere. They are important because they indicate not only the potential presence of life, but also its level of sophistication. It's important to understand that all biosignatures are really just potential biosignatures, because while they could be signs of life, they could also be caused by non-living things. A big challenge scientists face is battling all the false positives that arise. There's no official classification system for biosignatures, but it's useful to think about them falling into three categories: gaseous, temporal, and surface signatures. Gaseous biosignatures are direct or indirect products of metabolic activity. The most common manifestation of this is the composition of the atmosphere. For example, the presence of haze could be an indirect byproduct of a methane-rich world. This could tell us that a particular planet hosts microbial life similar to what we had on Earth over 2.5 billion years ago, before the Great Oxidation Event. It's similar to the example we explored earlier. of how having free oxygen in the atmosphere could be a biosignature for photosynthesizing life. Temporal biosignatures are time-bound changes that can correlate with biosphere activity. On Earth, the concentration of carbon dioxide in the atmosphere rises and falls with the seasons. Vegetation grows in the spring and decays in the autumn. This oscillation is way stronger in the northern hemisphere than the southern hemisphere because it has more landmass. In theory, we can use this kind of information to see where on an exoplanet life is most likely to be. However, nothing is so black and white. Temporal biosignatures can be caused by abiotic factors too. The concentration of methane in Earth's atmosphere, for example, changes mainly due to its interaction with water vapor in the troposphere. So, even though the methane itself is of biological origin, the temporal oscillations are dictated by abiotic factors. I personally find surface biosignatures to be the most interesting. Basically, every planet reflects some light from its star. Different materials on the surface of the planet will reflect different combinations of wavelengths of that light. This results in a unique reflectance spectrum for every material. Surface features like rocks, snow, water, and soil can all be deduced from reflectance spectra. Life can also influence the reflectance spectra of a planet. This is an example of a surface biosignature. Here's where it gets super cool. Today, the Earth appears blue from space because its oceans reflect a lot of blue light. But some scientists think that there was a time when our planet would have been purple instead. There's an ancient family of microbes that produce a protein called bacteriorhodopsin. Bacteriorhodopsin can create energy without a carbon source, something plants can't do. It literally just turns sunlight into metabolic energy that the cell can use. It is thought to be one of the simplest and earliest bioenergetic processes to have developed. But why would it make Earth appear purple? Bacteriorhodopsin contains a pigment that reflects a lot of purple light. This makes its cell membrane appear purple. An early Earth could have had oceans full of this organism, turning the whole planet visibly purple. While it's just a bit of fun to imagine purple Earth, the implications of this theory mean that these microbes and others like them could be a very useful surface biosignature of exoplanets. Of course, the reflection of purple light alone doesn't mean much. But if it were to be combined with evidence of a highly saline surface, that could start to paint a more convincing picture. You see, the specific family of ancient microbes that produce this purple protein is called halophilic archaea, or haloarchaea for short. Archaea are some of the most ancient lineages of life on Earth. Even though they're single-celled organisms, they are not to be confused with bacteria. The two are actually totally different domains of life, but we won't go into that. Halophilic means they grow and survive best in conditions of extremely high salinity. Halophiles belong to a larger group of organisms known as extremophiles. Extremophiles are life forms that are able to live in very hostile environments. There are lots of different kinds of extremophiles that are well adapted to different kinds of extreme conditions, like very high or very low temperatures, pressures, dryness, radiation, salinity, acidity, heavy metals, or any combination of those extreme conditions. These organisms span the globe, inhabiting the harshest parts of our world. From sulfuric hot springs in Japan, to the Atacama Desert in Chile, to sewage treatment plants in Germany, and even a hypersaline deep lake in Antarctica, where our beloved haloarchaea were found. What makes extremophiles so interesting is their unique biology. As you can imagine, living in such extreme conditions forces you to get creative. Extremophiles' biochemistry and physiology are often modified in very clever ways to help them adapt to their harsh environments. And this makes them an astrobiological goldmine. Many extremophile habitats on Earth are surprisingly similar to the conditions on other planetary bodies. These regions on Earth are called analogs, and they can teach us a lot about where we might find life beyond our home planet. For example, the Atacama Desert in South America is a very arid environment with high salinity, high UV radiation levels, and oxidizing soil. making it quite similar to Mars. In fact, we've been studying it as an analog to Mars for years. Life has been found across the Atacama Desert, but its presence is highly patchy. In a way, this is good news. It helps scientists identify the exact factors that cause life to appear where it does, and therefore, where we might have the best chance to find it on Mars. The extremophilic yeast Exophiala is another example of this. It was found in high-altitude regions of the Atacama and caught the attention of astrobiologists pretty fast. Researchers were looking at how Exophiala's protein expression changes in a simulated Martian environment. After the first 24 hours of exposure, they found some proteins were overexpressed and others were underexpressed compared to baseline levels. However, there were no signs of heat shock proteins, the compounds organisms release in response to high-stress conditions. Even more surprisingly, 7 days into the exposure, Exophiala's protein expression patterns were back to normal. This is a big deal because it indicates that Exophiala is able to recover and survive under Martian conditions. In other studies, Exophiala showed great resilience to the Earth's stratosphere. It also showed the highest UVB and UVC resistance when compared with other similar yeasts. All this together makes it a promising model for potential life on Mars. It may be a stretch to suggest, but it is possible any Martian life we might find could be biochemically similar to this humble yeast. Certain species of our haloarchaea are also being studied as exciting astrobiological models. Specifically, Halobacterium NRC1 and Halobacterium lacus profundi. They are both extremophiles that thrive in high salinity and are great candidates for understanding the potential life on Mars and Jupiter's moon Europa. Think about it. If there is a planet where conditions are totally different from ours here on Earth, it makes sense that the life we may find there could be completely different. But if the planet does have similarities to our own, is it really so crazy to think that life there might look similar too? Mars is a specific focus for our search for life because it has several similar early geological processes to Earth. Scientists have determined that Mars was possibly much warmer and wetter in its history. This suggests there could have been super salty brines on early Mars. Like we've said, these hypersaline environments are ideal for haloarchaea. Wouldn't it be cool if these ancient microbes had been enclosed in Martian brines, lying dormant all this time? Similarly, Europa is thought to contain salty oceans, 2 to 3 times the volume of Earth's below its inhospitable surface. This subterranean ocean might also be chemically rich, thanks to a silicate seafloor and surface oxidants, making it a great candidate for halophilic life. In fact, scientists have been testing this theory by shooting haloarchaea into space since 1994, testing the types of harsh environments in which they can survive. The first mission involved the ejection of a capsule into low Earth orbit. The short-term exposure results were promising, with the microbes surviving the initial exposure. The European Space Agency then designed the Exposal facility for medium and long-term exposure experiments on the outside of the International Space Station. 3 separate missions were launched in 2008 and another in 2014, testing the survival of various halophilic archaea strains in space. In March 2019, results emerged from the ESA's BioMex experiment. They reported that microbes from Earth survived 18 months of space exposure outside the ISS, suggesting life could theoretically survive on our neighbor Mars. Clearly, there is still a lot we don't know about these microbes. For starters, they've only ever been studied in isolation. How would they interact with the wider ecosystem? All the research so far has focused on sodium chloride. But how would these organisms fare in other salts? So even though they seem like promising candidates with lots of potential, more research is needed to fully understand their implications for astrobiology. So how does knowledge of these ancient microbes turned astrobiological models help us in the search for life in the universe? Well, within our own solar system, having intimate knowledge of these organisms can directly impact where we send our space missions. For example, the Spirit rover was sent to the Gusev crater to investigate a suspected ancient lake. We already had lots of evidence for volcanic hydrothermal vents on Mars, But Spirit discovered lots of new evidence pointing to hot spring activity in Mars's past. Hot springs are great places to look for biosignatures because they can preserve ancient cells in their chemical precipitates for a long time. It would be an ideal place to find the fossils of Martian organisms, if they ever existed, there. Beyond the Kuiper Belt, studying extremophiles lets us better understand how life arises and evolves in the universe. Firstly, it can show us what traits to look for in distant planets, a certain spectral fingerprint or a particular gas in the sky. It also indicates what the signals we receive from distant planets could mean about how hospitable they are. Extremophiles show us what types of life we might expect on other planets. And finally, they push back the limits of life as we know it to redefine what is possible, time and time again. Our searches for signs of life haven't been very fruitful yet, and will likely take many decades more. Designs are already in the works for a new generation of telescopes that will take to the skies in the 2030s and 2040s. The Nautilus is a concept telescope that could be operational by 2033 and would enable scientists to cover 50 times the light collection area of the James Webb Telescope, thanks to its large aperture. Another concept, put forth by NASA, is designed to observe potentially habitable exoplanets around Sun-like stars. The Habitable Exoplanet Observatory, or HABEX for short, would look for biosignatures like water and methane. It would also become the first telescope with the ability to directly image an Earth-like exoplanet. They sound fascinating. I hope they get approved. We've come a long way, and we still have a long way to go. But we are not giving up on our epic mission to find other life forms and their homes beyond the bounds of our planet. Our distant relatives will just have to wait a little longer to be found.